welcome to everyone here who has not heard the technical difficulties for the last 5 minutes. I'm talking about noise and artifacts in MRI and fMRI. I'll say that there's some other talks in this series that have also covered this topic. Paul Taylor's quality control talk is processing pipelines talk, particularly the quality control section. In the end, he goes through step by step a bunch of specific different artifacts. The high resolution talk talks about some artifacts that you particularly see at high resolution, but also gives you a sense of what MR acquisition artifacts look like in general. Barak's physiological confounds talk focused a lot on different types of noise relating to physiological confounds like respiration and cardiac and how to get rid of them. I'm giving a talk in a few weeks, which is about multi-echo fMRI, which is a tool for reducing noise, which can be used as a tool for reducing noise, and probably most talks in this series are covering this topic. So what I'm going to try and do today is just give a more higher level picture of what do we mean by noise in MRI data with some examples. So I think a good example of why this is kind of messy is this paper by Larry Wold and Jonathan Pellemani, where the title is Impacting the Effect of fMRI Noise Through Hardware and Acquisition Choices, Implications for Controlling False Positive Rates. So you think, they have the word noise in their title, it's going to be about noise. The first sentence of the introduction is applied to fMRI time series. The word noise is so non-specific and carries such negative connotations, it should probably be eliminated from fMRI vocabulary. So this simple word that we all know is actually a little bit more complex. So what is noise? What do we mean by noise? I have an audience, so I might as well have people like, what would you say noise in fMRI data is? Okay, it's gonna be that type of audience. So a nice dictionary definition is noise is irrelevant or meaningless data or output occurring along with your desired information. The one way I like to say that is it's literally the stuff that gets in the way of measuring what we want to measure. And I think that if there's any take-home message from this talk to get, it's that noise is defined by your study's goals. So and I'll give you some examples of that. And one study's noise can be another study's signal. So I think this is a classic example of that in our field. So this is actually PET imaging, but for a long time, you did these activity maps just like we do with fMRI. And for a long time, you had your areas of activity and you do a wide variety of tasks with PET. And Marcus Rakel and a few other people noticed that there were these other areas that whenever you saw the areas of interest, they seem to always have a drop in activity. And they sort of made a map of, hey, what are these areas that are, seem to be always, not our areas of interest, not the signal interesting, but always signal dropping across time. And this eventually became called the default mode network, which is a very active area of study now. This combined with another area of research, which was looking at random fluctuations in signal. So very often, we want to check the stability of the scanner. We want to check how things are going. You put someone in the scanner, you tell them to do nothing, and you're just collecting data where they're not doing anything you know about. That's, for a lot of studies, that's noise. That's just how much the data fluctuates over time. But Bharat Biswal back in the 90s said, if I put someone in the scanner and I tell them to move their, tap their fingers, you're going to see an activation map that looks like this. But if I put them in the scanner and I tell them to do nothing, and I just say, do voxels in one location correlate with another location, we see a remarkably similar map. So these random fluctuations have structure and meaning if you're looking at it in this like connectivity method. And we can put these two findings together and say, like, this random default mode network, which people had, was kind of like, that's interesting. And these connectivity measures, which is also nice, suddenly people started saying, hey, if we do correlations of these resting time series in the brain, this default mode network thingy seems to pop up a lot. And look, it varies among a bunch of different clinical conditions. So these two types of signals that a lot of people, at first glance would say are noise, turned out to be, for certain types of studies, a very active area of research. So that's sort of like my caveat going in, and I'll keep coming back to that in these applications, but I also want to take a step back and just talk about, when we're talking about noise, what are some measures we use? A common measure we use is signal to noise ratio, which is you take, typically in MRI, where we use SNR to refer to structural images. So you take some measurement of the sample of interest, like inside the brain. You take some measurement of signal outside of the brain where you don't spend signals, and you divide it. So, for example, in this case, this would be a high signal to noise image, a relatively high signal to noise image, versus a relatively low signal to noise image. So it's the difference between the background and the foreground and the image of interest are a little bit less. Another measure, and this term will come up both in this talk and a bunch of other talks, is temporal signal to noise. So if you're doing an anatomical scan, you want to see, like, do brain regions change? Does someone have, some sort of lesion? You're going to do structural imaging, and maybe that SNR is a good measure. But if we're doing fMRI, temporal signal to noise is a very common measure. And the way it's commonly used in fMRI is you put something in the scanner, like I said, you put something telling you to do nothing. And ideally, if it's like, a jar of oil, you don't expect to see signal changing over time. So if you take the mean signal in something where you don't expect the signal to change, and you divide it by the standard deviation, that's your temporal signal to noise. So you can see in this jar of oil, there are some areas with higher temporal signal to noise than others, and it's not uniform. If this is also a valuable measure in people's brains, but like, as I just said, if you do a task, you're causing variations, and there's other factors as well. And the last measure that comes up sometimes is contrast to noise. So if you're doing a task, you can have this temporal signal noise, but you can also have an effective interest. So if you're showing someone, any stimulus, a flashing checkerboard, you're going to see a response of, how large your response is to the signal. But if you repeat it many times, every time you repeat it, it's going to be a little bit different. So essentially, how much it varies trial to trial could be a measure of contrast to noise. So a common way to measure that would be, the magnitude of your response divided by the standard deviation of the variations over trials, or just a measure of a fit statistic. Sometimes we use the contrast to noise. Contrast to noise is also using in structural imaging, where you're saying, what is the contrast between, let's say, my gray matter and my white matter in the brain. So you can see in this image, there's a sharp boundary between gray and white matter, and in this image, it's a bit blurry. So that's an example of how you might use contrast to noise as a measure in structural. Any questions so far? Okay. So how are we going to think about noise reduction and noise removal? I'm going to divide this into, with a bunch of examples, into three general types of noise: unstructured random noise, structured random noise, and structured not random noise. So for unstructured random noise, let's use this this image as an example. This is no noise. We can add just random noise over the entire image. So in every single scan, you can still see the image, but it's just a little bit noisier. If you average 100 of these, it doesn't look like the original, but it's a bit more similar. If you average 1,000, it looks pretty close to the original. So if you're dealing with with structured noise, obviously the less noise you get in the original data, the better. The more times you repeat and you can average, it's going to reduce your noise. And if, there's always a big if, all data are similarly noisy, it's unlikely to bias results. So the most common place this type of noise comes up is sort of in acquisition decisions. So if you're, using a 1.5 Tesla versus a 17 Tesla scanner, you're going to get, less of this noise at a 7 Tesla. But if you're imaging really small, you're going to get more of this unstructured random noise. And the other thing to keep in mind is this is unlikely to bias results if it is even. So I'm going to keep stressing this again and again. So hypothetically, if you're doing a study and half your volunteers are on one scanner with one level of baseline noise, and another half are on another scanner with another level of baseline noise, or the same scanner, we do quality checks over time, and the underlying noise profile can change for a variety of reasons. It's still random noise, it's still unstructured, but it can vary over time, and depending on what your analysis, it can make a difference. So it is worth keeping an eye on. And this is sort of a good example of like how to think about this unstructured noise and averaging in a real example. So this is a task my colleague Javier did a bunch of years ago, and we're still using this task again and again, where we have some wonderful volunteers who we showed a 20-second flashing checkerboard to over we did, I guess, 5 times over 5 minutes. And they did this 5-minute task 100 times. They were scanned for 9 hours over multiple days. So thank you very much for our volunteers. And what that said is this kind of gets to the question of like, what does our data look like if we average enough? So this is primary visual cortex, where we expect to see the biggest response to a flashing checkerboard. And if you look at an individual run, that's what the signal looks like. You can just look at any given run and you see a pretty good response. If you look across all 100 runs, you can see they're different. That's our temporal signal, that's our temporal noise and our contrasted noise. But in any given run, you're going to say, yeah, they're responding to the task. Now, if we average, so this is just one of our runs. If we average across runs, you can see by the time you've averaged all 9 hours of data together, we get actually quite a good response. So this is an example of what we can see if we average enough and to remove this type of noise. Here's another, this is the exact same task. So this is a flashing checkerboard. There's no sound component to this task. This is what happens when I'm looking at a region in primary auditory cortex. If you look at the individual runs and you said, is there a task pattern? If you can see in those individual runs, that's very special. But if you average across those 100 runs, you can see that there actually is a pattern, that the signal is when the checkerboard appears, which is the blue lines, the signal actually goes down a little bit. And it's a much lower magnitude and a much higher noise level. So in the same task, in the same data set, there's one area of the brain where we have a very high contrast to noise, another area of the brain where we actually are seeing a significant effect with a much lower contrast to noise. So as you're planning and thinking about studies, it's not just sort of a universal measure. It's like, what are your expected effect size in areas and how much data do you need to get it? And very often, especially for task fMRI, we talk about, the areas of activation, but as you see, if you get enough data, it changes. So this is this exact same checkerboard task. These are the areas significantly active for 5 runs, so about 25 minutes, 40 runs, 65 runs, and 100 runs. So if in all our studies, we get all our volunteers to only spend 9 hours in the scanner, we would see essentially statistically measurable and significant responses across the entire brain. There's some follow-up studies that it's not 100% of the brain, but you are guaranteed pretty much whatever your study is. If you collect more data, you will see more things that are significantly active. So this again points to what is our noise and what are we measuring and what are we missing? Any questions on that before I move forward? Okay. So I mentioned first was this unstructured random noise. Now I'm going to talk about structured random noise. So let's say we have some sort of spatial artifact, and every single time you see the structure, you see these, obviously each of these look different, but they're all randomly different. So if you average them together, it looks close to the original. If you average even more, it's, you can't quite get it the same, but it's fairly close to the original. So just like the unstructured random noise, the less noise you get is better. More repetitions reduces noise. And again, if these data are only randomly noisy, it's unlikely to bias results. But this can cause serious problems if you're looking at individual data sets and not averaging. So, just to give you an example, things that could fit in this category of unstructured random noise are head motion, respiration, and heart rate, variation in neurovascular coupling, and trial-to-trial behavioral variability. But this all depends on what your task is and what your goal is. So in some cases, every subject moves their head. There's, some move more, some move less, but you have a one study, one population, it all averages out together and it's no problem. But that's not always the case. So these ideally cancel out, but and I would say just because people thought of these categories as things that should cancel out for at least the first decade, if not 2 decades of fMRI research, these were sort of not really worried about that much. It was like, you collect more data, they'll go away, don't worry. But like I said, now, that's only if those are unstructured. But we can also have those same signals that are structured by the noise. So we can have task correlated head motion, task correlated respiration and heart rate, and other unmodeled variation. And I'll also add on that as the field switched from task to connectivity, a lot of these become much more inherently a structured source of noise that are much harder, much more important to deal with. And I'll give some examples of what these look like. So one example is that, these noises can be task correlated. This is a paper a few of us wrote a bit back, where we try to always measure respiratory rates, respiratory depth and respiratory rate and a pulse oximeter to calculate pulse. And it turns out that you do a task. This RVT is a respiration depth, so essentially how deep your breathing is. And whenever the task, which are these little blue humps here, the person on average was taking deeper breaths during the task periods and not during the off periods. And if you attended Brock's talk, you know that respiration variations is one measure that we know causes global changes in fMRI time series. So this is going to sort of integrate itself into your data and be a non-trivial source of noise. I will say there are ways to remove this, and some of them work better than others, but whether or not you remove it, it's incredibly critical to know if it's happening. So I am a strong advocate for always collecting respiratory traces and cardiac traces, because at minimum, you can say, hey, my subject held their breath every time my task appeared, or they started taking deep breaths every time they appeared. Now I know that, and I can think about it and think about what to do. If you don't collect that data, you just won't know. So that was like breathing variation. And we also have things like head movement. So this is another study, which was, so frame-wise displacement is how much, on average, how much people are moving their head across a run. And each colored bar here is a different task or rest. But the bigger thing to notice is the patient population. So we have control, ADHD, bipolar, and schizophrenia. And what you can see is people with, essentially the control for every one of these colors, generally the controls have on average less head motion than these other groups. So if you have a measure that's going to be sensitive to head motion and you're studying two populations, that's going to cause trouble. So this is just sort of another example of the same data, age related for all these tasks, we're seeing more variation, more head motion across age. So if you're not watching for this, it's going to cause trouble. And I'll mention, one way people do this is we talk about resting state a lot as a measure, but it turns out that when you tell people to sit in the scanner and do nothing, they move their head more than when you give them something interesting to do. So even showing them a movie, or in this case, Inkscapes is like a glorified screensaver, just having something to look at makes people move their head less. So this is an example when thinking about noise. We can talk a lot about noise in how to correct for it after the fact. We can also talk about how to, reduce noise while we're collecting our data. So I just wanted to throw this in as one example. Before I go on, anything else any questions so far? Yes?
Will you talk about how to correct for it? Because I have task related motion, and then I thought regressing it out removes your signal.
So for task correlated head motion,
I mean the respiration.
For head correlated respiration. So how do you move task correlated respiration? Some of it I think was covered in Brock's talk. There's a bunch of different methods. I will say I've worked on several. None of them are perfect. The paper I mentioned earlier, this one actually discusses some variation. What was noticed in this paper is that the respiratory effect is on a slightly different time scale than your task effect. So it might be possible to sort of remove things that are at one leg but not another. There are other methods. There's a method called Rapid Tide by Blaise Federer's group and some other things, which are they're all regression-based methods that might help. The multi-echo talk, I mean, if you've already collected data, it's not use, can't collect multi-echo. But there's various methods that, like, I would say like any ICA-based method has the potential for separating a respiratory artifact from task-related something that's similar, because if they have a slightly different spatial pattern, it gives you the possibility, but it is hard. Like, nothing I said is gonna be perfect. So I think it's knowing your data and knowing your applications really comes down to it. And I also mentioned one of my previous talks, in my talk, I think 2 weeks ago, that I had this data set where some people held their, I was showing a stimulus for like 1, 3, or 6 seconds. And there were certain volunteers that just, they held their breath as long as the stimulus. And there was nothing I can do. There's absolutely no way I could have solved for that issue. And that's never the fun answer, but it's sometimes the answer. So here's another example of structured random noise. So instead of having these patterns, we can have, let's, to link directly, we're using MRI, we can have a dropout region. And that dropout region is in a different place each time. So no matter how much you average, there's still going to be something there. Like they all overlap, they're not quite the same. There's still going to be something there, and there's nothing you're going to do that actually removes it. So the more you can reduce this at acquisition, the better. No amount of averaging is making it go away. Sometimes, like with what you're saying, you can try to actively remove the stuff from data processing. And the other core thing is just knowing your limits from interpretation. So these are 2 examples of these types of noise. This image on the left is ghosting, or phase wrapping, where you're seeing essentially the same image wrapped over on itself. And sometimes with fMRI time series that you might actually get, like, so this is like the back of the brain looping over the front and the front of the brain looping over the back. And sometimes you'll get your time series interacting in those areas as well. So this could be bad. So this is something, if something is a really strong ghost, that might be a data that's almost impossible to use. But this is something to pay attention for at acquisition. And this is an example of distortion and dropout. So this is an area, the brain is a little bit distorted, and here's an area of the brain that just doesn't exist anymore. And this pretty much all fMRI has dropout. So depending what areas you're looking at, you're going to want to try and assess where they are, and also assess if that's how important that is to what you're doing. So going back to the exact same smiley face example, this is something that I think is happening quite a bit, that I feel the field is not grappling with as much as they should be. We have a lot of studies that, especially connectivity studies, you take the whole brain, you divide it into parcels of regions of interest, and then you average the signal for each region of interest. So let's say you turn the brain into 400 time series. And then that's what you do your analyses on. So you're doing correlations between those 400 time series, and making connectivity maps, doing graph measures, doing all this other stuff. We always have dropout in the orbital frontal area. But let's say in one case, our dropout fully covers this orange region of interest, mostly covers this one, covers a little fraction of this one. In this one, the dropout covers a little bit of the orange, most of these 2, a little bit of that one. Here it's covering another bit of the orange over here. So if we just take our data and average the usable voxels in each of these subjects, those regions of interest are getting, fundamentally different bits of brain tissue in the same ROI. So your same ROI might be measuring the anterior part of that orange region of interest in one subject and the posterior in the other. So you're looking like, a few million or different neurons firing that are completely distinct from each other. And I think what's concerning about this is because once you've collapsed and averaged, this is hidden from the experimenter. There's really no way to say, what am I looking at or not? So this is analysis step you really have to sort of be aware of and say, hey, this orange region of interest is just not reliable across my population. Maybe I just have to remove it completely. And a lot of people definitely do this, but I think it's something that is meriting more discussion. Yes?
So you have a proper structural image.
Yes.
Most idea from where I'm here, T2 or T1 image.
Yes.
And you do some form of API distortion correction that may help with this,
Yes.
or do you just discard the data?
So you can definitely do distortion correction. So there's two different problems. There's distortion and dropout. So distortion, like you can't, there's some really bad distortions that you can correct, but we definitely have methods that reduce distortion. But dropout is essentially your signal, because of various artifacts, is just decreasing so fast that you just don't have signal. So if you don't have signal, there's nothing to bring it back. There are acquisition methods that will reduce your dropout or change where your dropout is. So like if, let's say, you were really interested in the amygdala, which is an area that sometimes has a lot of dropout, then at the acquisition stage of a study, you should spend a lot of time saying, let me make sure I'm getting amygdala signal as robust and reliably and fully as possible. Or I think what happens actually more often is if you download a data set where you're studying that they were happy with, whoever collected the data was very happy with, but they didn't care about the amygdala. And you care about the amygdala, and suddenly you're looking at this data set, you have to, give a good hard look at it and say, I can't use this data because they did not get reliable in this amygdala signal. Yes, in the back?
Is there any reasons to like bootstrapping? Let's say almost under.
Can you speak up a little bit?
Sorry, yeah. Is there ever any reasons to like bootstrapping? Let's say you have samples where there's relatively low dropout in your knowledge, kind of region of interest, but about 30% or something like that are missing in June.
So the question is there any way to sort of address this with bootstrapping? And I would say completely depending on your application. So absolutely, there's certain cases where you can model what's missing. You could also potentially say, I'm not using regions in one study or another. But it depends a lot on the study. So like, if you have a study saying, I have 5 regions I care about, and I'm studying the connections between these 5 regions, you could say, in 20% of my subjects, I'm missing one of these regions and I remove those connections. If you're doing a method that essentially involves whole brain connectivity, so like a graph, like there are these graph theory measures, which bootstrapping is often done with, but if a certain number of your regions of interest are missing in a certain fraction of your subjects, that's going to affect all the measures everywhere. So I think there are ways to use bootstrapping to essentially assess the extent of the problem and possibly decide, and then that'll help you with interpretation. But it's not, I would say bootstrapping is a tool for interpretation. And you can use bootstrapping to say, how is this affecting my results? And it could be that you do it and you say, like, it's not affecting my results, but I would frame it in that way. And just to give an example of this dropout, this is fully speculation on my part, but I think this is a study that's been pretty prominent, so I think it's fun to speculate about prominent studies that they were trying to. This study was claiming that if you're doing these resting state connectivity measures, it takes thousands of subjects before we get a stable measure, and that a stable measure for like behavioral trait measurements. So in this case, they were looking at this metrics of psychopathology traits. And they said, these are the regions of interest that are involved in that are most significant in these traits. You'll notice a bunch of these are in areas that are known to have dropout. And they're saying it took, before our correlation values got stable, took a huge number of subjects before we got individual measures. But hypothetically, if these regions of interest were literally measuring different amounts of cortex, different places of cortex in each person, that is a measurable and correctable area of noise that could be fixed. And I will stress again that I am completely speculating and picking on this study. This may or may not be at all, but I think the reason I'm bringing this up is when you look at a study like this, these are good questions to ask of like, what would happen if they did or didn't model dropout correctly? I also mentioned a little bit about hemodynamics and vascular bias. So we know that the hemodynamic response shape, so that's the vascular pattern that responds to different people, does change across various factors. So like this is a study I did a while ago where I was using an exciting flashing checkerboard with a younger and older population, and I showed that if you did that the signals magnitudes were lower in an aging population across multiple regions of interest. But I also showed that if we did sort of a vascular challenge, so a breath holding task, we saw a similar pattern in a lot of areas where the signal also dropped. So that says that this might not be, your visual cortex is responding less as you age. It could be that your hemodynamic response, the way your blood flows through your body, is altering at different ages. And if you divide, you can actually see that these changes actually are retained in some areas. So this does imply that we are getting some signal, but this is just another example of like the types of biases to potentially look out for. And there's a whole bunch of other calibration measures. I think Brock, you talked a little bit about this last week as well. And this hemodynamic variation issue comes up a lot, especially when you're talking about temporal modeling. So there's a bunch of different methods like dynamic causal modeling, which are essentially saying, given assumptions about the shape of this hemodynamic response, can we predict when one event happens before another? And this, I'm not going to go through this picture in detail, but the basic idea is this is a method that tries to sort of assign causality. But what I did here was I said, instead of saying I have a hemodynamic response where I shift the peak, I said, what happens if so hemodynamic responses tend to go up and then below baseline and return to baseline. So I said, what happens if I just make that drop below baseline a little bit bigger? And that violated their model of how they assume hemodynamic responses vary, but they do. It's something that I see quite common, and their model broke down on causality. So this is one of those things that if you're studying something like causality, knowing how something like hemodynamic responses can vary is critical for interpretation. So before that, the next little section, any questions right now? Okay. I've hinted at this a few times, but this is a good example of like why things get a lot more challenging with connectivity measures. So there's other measure tools we use, but the most common tool we use for measuring connectivity is correlation. So you take two time series, you correlate them to each other, and the more correlated they are, the more similar we say they are. So here is a case where, let's say I have two time series that are, I scale them differently, but they are identical. So the R value is 1. And I have another time series, which is different, and the R value is 0.05 between the green and the red. I'm going to add the scanner-based noise, which we always see. This is just kind of random noise. I'm going to add cardiac and respiration noise, which has temporal structure to it. And I'm also going to add a spike because they move their head at some point and we got that signal of data. So now what do we have? We have the two time series that were initially identical without noise. Now they're still highly correlated. So the R.5 is pretty much significant than anything we do. But these other two time series are at R equals 0.31. So if we're not accounting for, different sources of noise, in this case, the respiration cardiac and the spikes, things are going to end up in our data that we don't want to see there. So for model, a lot of these things like respiration cardiac noise, unless you're task lock breathing, is annoying. But when you're talking about rest, if you don't account for this stuff, you can get in a lot of trouble. Yeah, so pretty much like this can contaminate results. So this is an example of one case when this happened. This was a study from back in 2009, where a group of Damien and Farah and a bunch of others made these graphs of different brain connections over time. And what they showed is that the same connections change between age 8 and age 13 and age 25. So these are two networks, and you can see how closely these regions correlate, have a clear age-related effect. And it talked about this change from local to distributed organization over time. The trouble is they realized, like as I mentioned earlier, younger kids move their heads more in the scanner. I know it's sometimes shocking, but yes, younger people tend to wiggle more. And if they measure, if they essentially remove the areas of high motion, so they essentially unscrub, so this is the data they presented. And the correlation between the children and adults was 0.56. So that there was non-trivial difference between these network maps. And after they removed the head motion, it was 0.7, which is, about as highly similar as we can get with noisy MRI data. So essentially, when they accounted for motion and removed the high motion images, their effect completely went away. And I love this quote from one of the authors, Steve Peterson. It really, really, really sucks. My favorite result the last 5 years is an artifact. And I love this. What I really love about this is this, this is good science. This is, you put a lot of work into something, you publish it, you're proud of your work, but you don't give up on it, and you come back and you realize that you made a mistake and you own it. So the reason this now a bit older, this is 2009, 2012, but the reason I keep coming back to this is when we're doing science right, this is what's doing science right looks like. So I keep looping back on topics with different examples, but why is noise removal so hard to solve? I think the biggest thing is that really the signal and noise versus noise is really defined by our study's goals, so that we can't. There's some types of data and some types of science where our definition of noise and signal are a little bit more common across a wide range of studies, but in what we do, it varies so much that we have some methods that apply and are well used. I think Paul Taylor talked about the AFNI processing pipeline, which gives best practices for a lot of studies, but there's so much that varies that you really have to be aware of those differences. And what's annoying noise for one study essentially biases all your results in another. And there are some factors that are measurable, but then that just because we can measure and see a problem doesn't mean you have a solution. And there's other things that aren't as easily measurable, like this, neurovascular coupling changes across populations or non-task specific behavior that you might not have to measure. Okay, I'm going to jump to a little bit of a slightly different section. Any questions before there? So I'm going to talk a little bit about, I'm not going to go super into depth about every types of way we can minimize noise, but I'm going to try and give you a few examples to sort of how to think about these issues during data acquisition. I'll start with sort of the obvious thing. We want the best SNR, best contrast to noise, best temporal signal noise, fewest artifacts, less distortion, best temporal resolution, best specificity, but we can't do all of that. So really what we have to do is balance priorities, and that varies by studies. So especially if you're doing data collection, it's critical sort of at the pre-acquisition phase to figure out what your priorities are. Do you care about your response shape, as precise response shapes as you can? Do you care about anatomical accuracy? So is distortion going to destroy your results, or is distortion something you can live with? So if you're doing layer fMRI where you want to know things down to the millimeter, you really need to know where things are. Or if you're studying a very precise area, you need to know where things are. If you're collecting 3 millimeter voxels and you're smoothing to 6 millimeter voxels, it doesn't really matter as much if you're a millimeter or two off. And how robust are your artifacts, and how robust are your artifacts that can bias a study. And I think the core message is sometimes what's optimal isn't obvious. So the example I'm going to do for that is a study on the flip angle, which is just an example of one of these parameters that most people who've collected data just sort of copy from whatever's there. But the flip angle essentially affects your signal to noise. So at the optimal flip angle for a scan, it is maximum signal to noise. But what it turns out is because it's affecting the signal of both what you care about and also various artifacts like breathing and respiration, others, it actually scales. The temporal signal noise actually scales with flip angle. Sorry. The noise, the magnitude of some of these physiological artifacts scales with flip angle as well as the values of the things you care about. So what that means is even though SNR is very sensitive to flip angle, your temporal signal noise is actually flat over a much wider range of values. And here's what this looks like in practice. This is another flash and checkerboard task. If you look at the Rohrbold responses, that maximum flip angle is going to give you the biggest response because that's your SNR. And as you drop your flip angle, the magnitude of the response gets smaller and smaller and smaller. But if you scale for this magnitude difference, what you can see is that the contrast to noise, so sort of how much they're wiggling over time, is nearly identical over all of these. So for a wide range of flip angles, even though we're getting bigger signals, it doesn't actually matter that much. And like I said, this is something that like most people don't have to think about flip angle, partially because of this, but it's just an example of something that, you know, at first glance, SNR is good, but it turns out it doesn't matter as much as you might think. Renzo in the other talked about, you know, high resolution, but just to frame this in thinking about acquisition, the smaller your voxels, the worse signal to noise you get, but the more anatomical specificity. So if the goal of your study is to see a difference between layer 2 and layer 5 of a cortex, no matter how large your signal to noise is, you're never going to see that with 3 millimeter voxels. So you actually can get more precision and more signal with smaller voxels for specific questions. So this is just an example, like even the pulse sequences vary. So this is just an example of there's 2 different acquisition methods, 3D echoplanar imaging and 2D echoplanar imaging. And you can see with larger voxels, the temporal signal to noise ratio is actually better for 2D, but when you're crossing over into like this like 1 millimeter cubic voxels, those 3D sequences do a bit better. So, and here are just sort of examples of what that looks like. So your 3D is actually, you know, at 1.5 millimeter voxels, you're getting a more robust signal with the 2D sequence. And at 0.75 millimeter voxels, you're getting a more robust response with the 3D sequence. What I would say for here is like this type of stuff that like, if you're designing a study, specifying your questions really well, and then going to someone with expertise in the MRI physics and acquisition is going to be really productive. So the more you can say, this is what my goal is, the more someone who has the skills to sort of define a pulse sequence and help you with that is going to be able to give you the best help you need. Similarly, there's different pulse sequences. So most of fMRI, we talk about gradient echo bold, which is skewed towards large veins. But other things that I think Renzo talked about, vaso is much more skewed to smaller vessels. So this is just an example of thinking about, again, these different pulse sequences, and depending what you're looking at, there can be different benefits for each one. Any questions for now? Just like I was talking about voxel size, the same is true for sampling rate. So when you collect faster, you're generally going to drop your SNR, and you actually are probably going to drop, for any single value, you're going to drop your temporal signal to noise. That's for 2 reasons. One, the more you're exciting the proteins, protons, the less signal change you're getting between each excitation, and that's reducing your signal to noise. And then the second factor is the ways we scan really fast generally are making sacrifices on your signal to noise. But you possibly do get better signal because if you're getting, let's say, 4 volumes in 2 seconds instead of 1 volume in 2 seconds, you have 4 times as much data, and that can benefit you as well. So there's this balance with a lot of temporal signal to noise. There's also a potential benefit that if you scan fast enough, you have the potential to just filter out high frequency artifacts. So this is an example of someone who's scanning at a 367 milliseconds TR. And what you can see is right around 1 hertz, this is like your cardiac. So if you could say, I have a clean cardiac signal around 1 hertz, you can design analysis methods that just filter that out, you know, really efficiently. The one thing I'll note there is this group by Laura Lewis showed that it actually takes really fast TRs to truly filter it out. So there's a bunch of people that have been scanning around 800 milliseconds or 700 milliseconds. And with the idea that, hey, you could filter this stuff out, it turns out that that's not enough to really pull out heart rate. So if you want to pull out heart rate, you have to go more like 400 milliseconds. And then to do 400 milliseconds, you're sacrificing a lot of other stuff. So then that kind of goes back to your acquisition questions of what's your priority. The other thing is, like, we get the better signal, but in the end, our human responses are slow. So they generally peak at around 5 seconds and resolve even slower. So you're not generally going to see faster stuff, even if you collect a super fast TR. That said, I will note that there's a bunch of really awesome work showing that with really specific, well-designed task designs, we can see things much faster, even with the dynamic response limitations. So this is a great review article by Jonathan Palmani and Laura Lewis. And if you have goals of seeing things faster, there's ways to do it, but it involves, really thoughtful task design to do it right. Another thing I'll mention is, so far I've been mostly focused, besides respiration and cardiac, I've been mostly focusing on MRI measures. But when we're talking about noise, there's other things we can do to keep track of stuff. So if we're using an eye tracker that can measure pupil diameter, how much they blink, how much they move their eyes, on the basic level with no noise removal, it's good to know if your volunteers' eyes are closed, because if you want them to do something and they close their eyes, that's noise. But this is a study by Sharif Kranbar, where they're doing a task, and in the interstimulus interval between tasks, they were looking at the responses, whether someone's pupil dilated, whether they blinked, and whether they had microsaccades. And it turns out that they were seeing different response maps depending on what their eyes were doing. So that is just one example. There's a lot of stuff on eye tracking. That is one way that if you're modeling more signals like eye tracking, even if that's not necessarily the goal of your study, it gives you one more way to understand what your volunteers are doing and potentially remove things that are either biased or unbiased in noise. Here's another example of the same thing. Especially for resting state scans, you put someone in the scanner and you tell them sit there and do nothing, but they're not doing nothing. They're doing something. And it turns out, so that in this study, for the data set, after the scan was finished, the investigators asked them questions about what they were doing during the scan. So there was about, I think, 15 questions or so. This is an example of 3 of them. So they were asking, like, were you imagining images in your mind? So, like, just picturing things in your head. Were you thinking about your surroundings of the beautiful scanner board in front of you? I'm editorializing it, I'm beyond the questions. Or were you thinking of like past events? And depending how people answer these questions, it turns out that there's variations between the network connectivity patterns. So this is, especially for resting state studies, there's a lot more you can do with behavioral monitoring, but just as a basic level, rest is not rest. People are doing things, and the more information you can collect about what they're doing is going to help control for that. So in some studies, this might just, like, going back to some of my earlier topics on sort of what's structured and unstructured. Some studies, this is just random noise. One person was thinking about yesterday, one person was planning a day in the future, one person was counting the number of rivets on the inside of the scanner board, or that just a little bit of variation in the scan, that doesn't matter. But let's say you're studying across populations, and a small child in there is constantly thinking about, when is this done, when is this done, when is this done, and someone else is planning the dinner they're going out to later that evening. That's going to cause a population difference. If you're not tracking this information, that's going to be a serious bias in noise. So I've kind of gone through all my slides. My main take-home messages, which I've repeated a bunch of times, but I feel like just one last time. There are some things that are noise in every fMRI study. So if you get more dropout is bad. If your data has, smaller voxels are gonna make your data look a little bit fuzzier. You can have, signal drift that you don't want. There's all these things that are noise everywhere. But the difference between in a lot of cases, the difference between signal and noise is often study specific. And even for these things that I just talked about that are always noise, their importance varies. So sometimes that, as I mentioned earlier, dropout in the amygdala is like, hey, I'm not studying the amygdala, who cares? And so, or I think the more true example is, hey, I didn't collect data from the cerebellum. Who cares about the cerebellum? So there's a lot of studies that don't have cerebellum, and for a lot of studies, that's not a big deal. For studies that think the cerebellum might be an important part of the brain, that is incredibly critical. So that's an example of some things might matter to some people more than others. And at the core, clearly defining what you think is your signal and giving deep thought to what sources of noise end up in your data is what's going to improve study designs and analysis. So that is it for my slides. So I'm happy to keep discussing and taking questions as long as people want. Yes.
So you were saying about high frequency artifacts. Can you, there were one slide that you say, that applying people have, fast enough, they
Yeah.
will fast enough around the high frequency artifact.
Okay.
So, you mean this artifact in a ham signal or in capsulate or is it I just don't understand that.
Okay. So the question is, what is I referring to by high frequency artifacts and how to remove them, I think?
Yeah.
I'm repeating for the microphone too, so it's the people, so it's recorded. The most common example people talk about for high frequency are your heartbeats cause a systematic variation in the signal. And your heart is beating somewhere between 40 and 100 beats per minute while you're lying on your back in the scanner. So, typically around 60 beats per minute, that means it's a 1 hertz artifact. But, and then you have respiration signal, which is, relatively high frequency. So that's going to be your breathing cycle is probably a breath every 3 to 6 seconds. But so these are adding quite a bit of variation in data. So like I'm actually analyzing correlations between respiratory and cardiac traces and MRI data right now, and the R value is around 0.2. So a fifth of your data are these respiratory and cardiac artifacts. There's a study by Monica Biancardi from 2007, 2009 or so that sort of gives one map of these percentages. So these artifacts are in the data, and they're temporal artifacts, which I think was part of your question. And there's a bunch of methods that sort of try to systematically say, what is the shape of this artifact and how do I move it? But if you sample fast enough, you can just say everything above, everything above 1 hertz, just get rid of completely, or 0.8 hertz. So like, just get rid of everything above 0.8 hertz. We don't expect to see anything we care about there. And we can get rid of that. The trouble is, I don't know your background on signal theory, but if you're undersampling your signal, it kind of folds over into lower frequencies. So if you're taking a sample every 2 seconds, an event that's a 0.5 hertz, if you have an event that's happening at a 1 hertz rate, it's going to fold over into lower frequencies, so you can't just filter something out. So that's, and it turns out that to really get rid of that cardiac artifact, you probably have to go down to like something around the 400 to 500 millisecond sampling rate.
Well, with that artifact, can you measure what's your heart rate or
Yes.
so synchronize with your data?
Yes. So there's a bunch of methods that with slower sampling rates, we can address it. So you definitely measure heart rate. There's methods that are used as essentially, a large sinus or a large vein region of interest to get the base time series. And these methods work. I use these methods, they work, they're not perfect. So if your goal is to reduce the noise, there are some excellent methods out there but if your goal is to completely eliminate the noise, I don't think any of the methods, truly eliminate it. Okay. Thank you so much. It was good to have a nice size room here.